Hi everyone, and welcome again to another Saturday live stream. I'm Oliver, and I'm happy that you could join me again. What have I planned for today? Well, as you can see over here, I don't know if some of you remember, but this is a mini terrarium in a jar that I made, also in one of my live streams here together with you several months ago. And it's not looking nice, as you can see. I mean, it's pretty bad. I mean, there was actually quite a bit of nice moss in here. You can see maybe a small over here, a small a piece of wood, some rocks, and I wanted to make it really look nice. And this is basically what's left over. And of course, I'm going to put it under the microscope. And of course, I've also prepared a couple of other specimens. Some of them we've already looked before, and I just would like to see how they have changed over the months. Last week we did quite a bit of a deep dive and did some staining of bacteria, but today's session is going to be a little bit more loose and varied. So I've got a whole list of things that I would like to look at and to show you. As always, I would like to say hello all around the world. I'm now not going to go through the chat like in the previous months because I want to save a little bit of time. But if you have any questions, please feel free to write them in the chat. But before we actually get started here, I have to talk about the so-called the elephant in the room. I think you say it in English. And that is maybe some of you have already noticed that my website and the forum have been down for several days. I was very busy the last couple of days getting it running again. And the website, if you visit microbehunter.com, you're going to see it doesn't look very nice. Because I'm going to completely rebuild it right now. I had a problem, as a matter of fact. The website and the forum were attacked. So basically, within a short time, millions of bots tried to scratch the contents of the website, and then basically it was shut down by my hosting provider. Essentially, it crashed their database and their system, and so they had to take it off. And then I had basically had to try to fix everything. It took me, I don't know, a couple of days, but now it's kind of working again at least. I also had to add some additional security here. So just in case you're wondering why there is sometimes when you visit the forum or the website, what is this screen? It's checking whether you're actually a real human being. Yeah. So this is the thing. Yeah. I'm going to, therefore, I hope that this is going to be stable right now, but I was told it's not 100% of a guarantee. Yeah. I'm also using AI to build a website. I am using this as well. And another short piece of information that I want to say is because the newsletters that people subscribe to were also hosted on this server. I could not even write newsletters anymore, because this was taken down. I mean, I had all of the email addresses, of course, backed up. But what I've done is also for the simplicity of use, I changed newsletter provider, and today and also tomorrow and the day after tomorrow, I'm going to send out the April newsletter again. Some of you might have already received it, but there is a certain limit of emails that I can send per day, and for this reason, it's got to be kind of distributed over a couple of over a couple of days. So I'm going to that's basically a little bit a short editorial here. I see that several people have already joined in. If you have any specific topic question, on topic or off topic questions, please also indicate this by saying @Microbehunter at the start of the chat, because sometimes people chat with each other. That's fine. But then I have a difficult time to actually find the questions which are directed to me. And for this reason, it's easier for me to find it. I'm going to basically make a stripped down version of the website, and I'm going to try to make this a little bit more efficient this way. So that is one thing here. So we're going to be having a look at this here. I made myself another sheet here. I also want to, but you know what I'm going to do is I'm going to start off with something different. I'm going to start off with a failed experiment. I just would like to show that to you, and I'm just for 5 minutes or so, because what I have here is over here. It is a little. I was really hoping. I'm not giving up. But I was really hoping that this is gonna work, and it did not work. So I need to explain this to you. What I have over here is a slide with pollen, and I got the pollen from these. from here. These are called the pollen baskets from bees. A friend sent them to me. So basically, this is how bees collect pollen. They're very compact, yeah, and this is how the bees carry the pollen back to the beehive. I took a small amount here, and I not only put it on the slide here, but I also mixed myself a sugar solution. So this is 10% sugar, and I added a tablet of boron. So this is basically a nutritional supplement. Boron, and I was I was kind of hoping to see pollen germination, and it did not work. I think that the reason is because these are not fresh pollen. I'm going to show you in any case a lot of pollen, and so that's just a slide with pollen without cover glass even. But my hope was a little bit to see the germination, but this unfortunately did not work. So what I have to try is now it's springtime in Central Europe where I'm living, so there are going to be more and more plants with pollen right now. So you see all of those little circles here, these are all pollen. And essentially when you add the sugar solution, what should happen is they should start to grow a so-called a pollen tube. Yeah, I was unfortunately not able to see that. You see the pollen essentially are still as they used to be. So that's basically how it looks like. This is now without a cover glass because I wanted to make sure that enough air is able to reach those pollen. It's been on here now for, I don't know, 2 hours, 2.5 hours maybe, and still nothing's happening. So essentially what I hoped is that there is this small tube growing out of the pollen, nothing like that. So what I'm going to do now is just for a few minutes, I would like to really go in and observe. When I'm going to add a, cover glass, does the BX53 beep when turned on? Let me try. Off. Yes, it beeps. Yeah, I don't know if you heard it. It makes a tiny beep, when it's turned on. I don't know. Did you hear that? So over here, that is again the desk view over here. So let's add a small cover glass. Yeah, so that we can go up with the magnification a little bit. Otherwise, it's going to touch the. Here now we see the pollen. Actually, they do look kind of pretty as well, but because they are kind of old already, and also when they're compact and when the bee collects them, then of course they might not be active anymore. Yeah. The beep is that the beep of the microscope is not very loud. Yeah. So generally, I think I like pollen. Obviously, they're nice to observe, but. So far, I was only accidentally once could I find a couple of pollen that were actually growing those pollen tubes. Yeah, and so essentially, I can actually show you how this would look like. Yeah, here's an arrow. So if we take this pollen grain there, then something would start to grow out of the pollen, and this then would contain the gamete. Yeah, basically the sperm cell to fertilize the ovule in the flower. Yeah, unfortunately, it didn't work. So that's actually one of my failed experiments. So to say, I was really hoping to be able to show you some of these nice pollen tubes. The recipe says you have to use a 10% sugar solution, and usually it goes better when you add boron. But in my case, it was not very successful. Yeah, so really, a pity, but I'm not giving up, and maybe in a future video I'm able to show that to you. But I decided I'm going to share with you in any case all of the successes and failures that I'm encountering here. Yes, exactly. As a matter of fact, ever smaller, Jesse is saying that for future stream, collect some fresh flowers. Today I went out to collect some pollen, but they were not mature yet. So it's still a little bit too early in springtime here. Yeah, so this is a little bit a pity, but that's just the way it is. Yeah, and it's part of the hobby, so to say, so that not everything works. Yeah, and for your info, what I'm doing is when I have a slide here, I usually put it in here. I put a lid on top so to slow down the evaporation. But essentially we can throw this slide away. Okay, so that's not needed anymore. So that is number one. Number two, something I wanted to show you, was it 2 or 3 weeks ago? yeah, I made this slide over here. If you remember the little carpet beetle, it molted, and I put some of the exoskeleton, basically the shed outer part. When it's molting, it's kind of shedding off its skin, and we made a mount here, and it's actually looking pretty good. It's actually a very nice mount, and I would like to show it to you because the mounting medium that they used, Uporol, is actually quite good for these things. Yeah, and I'm going to go bright field here, and it started to clear up even more. And this is how it looks like. So that's basically the exoskeleton of the carpet beetle, the larva of the carpet beetle, and yeah, lots of hair. And when I looked at this, this over here, what I think what those two dark dots are, I think, I don't know, correct me if I'm wrong, they look like the mandibles, the mouth parts. It's kind of, I just discovered this today. Those things here, yeah, seems to be, looks a little bit like the mouth parts of the carpet beetle here. Yeah. Yeah, so exactly, now we can go really close, yeah. Yeah, yes, the Uprol cleared those parts quite nicely, yeah. And that's much brighter than before, and now we can really go in, yeah, because at least on the side the mounting medium has already dried. So, and we're going, I don't know, we just pick something bright over here, yeah, and we can really go in. And one of the things is of course, oil immersion is not possible with permanently mounted slides, because then of course, yeah, it's going to smear, the oil is going to be all over the slide. Yeah. So I'm quite happy with the result. Maybe some of you remember from a few weeks ago, there were still a couple of bubbles. The bubbles are also gone now. Yeah, so look at this. You can actually see quite nicely where the hair actually starts to grow out here. This is really, yeah, really nice. So I'm super happy with that. I think it still might not be entirely dry. There is usually some pie times thumb, as we say. We have to wait about 6 weeks until the medium is completely dry, depending on the amount of mounting medium that you're using. Yeah, but it's generally quite, yeah. You want to find a clean of the old one arrow hair? Yes. Where is it? Let me go down a little bit. I have to find them. There were plenty of them. Here's one. Yeah, because exactly they have those weird looking. Here they are. They have those weird looking. Yeah, arrow hair. Yeah. Basically, setae they're called. Setae is a general term that we refer to when about the hair of insects are called setae. It's a general term. Yeah. Maybe some of you have already look at the top here. I'm now using my 60 times objective, and what I'm doing right now is I'm turning this ring here. Yeah, there's this adjustable ring here, and this ring, by turning it, you are able to adjust for the different thickness of the cover glass. Yeah, and you can actually turn it until you get them. Yeah, there's not a huge difference. Yeah, but this is kind of for adjusting the cover glass thickness. Yeah. I better not forget to label it. You're gonna laugh. I mean, I actually had it lying around here with a couple of other slides, and then I already had problems. Yeah, I had to check a few slides until I found it. Yeah. So very nice sample. I'm happy, and generally quite happy with the result here. Okay. So we're gonna move on. Yeah, I just want to show that to you, and I think we're just now going to start putting some of this horrible looking, how do you say this thing here under the microscope. I did try it a couple of I think 1 or 2 weeks ago. I tried it very briefly as well, and there are, of course there is some nematode worms and so on can be found in here. Let's give it a try, and let's see what we're able to find. It smells like. It smells, interestingly, very similar to slime mold. I mean, you remember some time ago I was growing some slime molds, and they had this peculiar smell, and this it smells the same way. Yeah. So not surprisingly, because maybe they're producing the same waste products. So, and what we do is the following. We take a little bit of yeah, it's difficult for you to see that. We take a little bit of, I always do this, I take a little bit of water, put it on the microscope slide here. And now we take some of this disgusting looking thing here from the wood, and we dip it in the water. Gee, it absorbs the water. And I really would like to know what's in here. So mostly decomposers. A little bit disappointing that the moss that was growing in here, yeah, is not really. I just returned this. That the moss is really not, yeah, looking very nice. It's also kind of decomposing and everything. It's a pity, yeah. But maybe it doesn't matter. Maybe that actually makes it interesting. So let's put this away. I don't even know where I put my lid. Yeah, I'll put it on later then. Cover glass goes on top, and for those of you who are maybe I don't know if there are lots of new people who joined in, but the format here is a little bit like this, that sometimes even I don't know what to expect. Yeah, so what's these labels you speak of? Maybe I can answer this. Okay, the problem that it's a little bit of a sloppiness from my side, and that is that the slides that I have here, right? I have to, yeah, I make those slides, and then I should add a label over here with a date, so it's easier for me to, yeah, remember when I made the slide. And I've been a little bit sloppy with that. So it's become a little bit like a running joke inside this live stream, because sometimes I'm so confused because I don't know what's on the slides, because I simply did not label them. I was simply too lazy to label it. Yeah. So that's essentially the thing. Yeah. So sometimes also my jars have some alcohol and some water, and then I forget which one's which. So bad laboratory practice. So let's have a look here. At least something that's moving. Yeah, of course, a nematode worm, and something looks like I don't know. Is it a rotifer? Okay, that's kind of weird stuff. Okay, but at least we can see that there is something moving. So I'm happy, because people like to see some moving stuff. Same as I. Oh, that's way too dark. Okay, yeah. So usually, yeah, if you're into hobby microscopy, what you always very often hear is online. Yeah, I like to observe water samples and so on. Of course, water samples are nice, but you don't always need water. You sometimes decomposing material is good enough as well. Yeah. It's a little bit very dense, the whole sample, but yeah, rotifers. I mean, I'm always kind of surprised that. Yeah, essentially they're able to, yeah, because there's only a very tiny amount of water on the wood, that this is actually evidently still enough for them to catch food. Yeah, stuff whizzing about. Some, yeah, it looks like an egg here in the center. Let's go down a little bit with the. Yeah. Let's just give it a rough scan of what we can see here. Yeah, of course, mostly worms. Not so many ciliates I've seen, interestingly. Worms and rotifers. So essentially, that might be essentially a little bit of biology. Mentioned already before. Are those what are they? Are they actually microorganisms or not? And those rotifers, yeah, and those nematode worms. Should we count them as microorganisms? And actually, per definition, and I know this is kind of a little bit of a language issue, per definition, these are not really microorganisms. They are actually referred to as microanimals. Yeah. So that is, yeah, they're referred to as microanimals. And because they're multicellular. So actually, yeah. So this one here is the round thing that you see here. That is the shell of an amoeba. Yeah. Acella, usually, or cella, that's the name of, yeah. So you see this round thing in the center here, that's, yeah. I have to tell you, it's kind of difficult to find amoeba. I mean, if you see those shells, then you know that amoeba are going to be around somewhere. But they're difficult to see because they move kind of slowly. Yeah, so So that is a little bit, so you can, that's basically what I like doing. I just like to observe. I am not so much into, I mean, there are some folks out there who are really much into taxonomy and into the identification of, and while I do have an identification book, I'm more into the observing part. So what is this guy doing? This Rotifer attached itself to the surface and it's now spinning around. So it's kind of fun to observe these things, but you know what? Let's do the following. Let's change the color a little bit. Shall we change the color? Yeah, let's go like this. Yeah. And let's have a look. Here we go. Let's open it a little bit. Yeah, that's an interesting one as well. Usually the amoeba, as I said, they move quite slowly. But when you have them time lapse, then it's can be quite interesting. So maybe we're lucky, maybe we're able to find one or the other. Yeah. Amoeba are, per definition, they are microorganisms. They're single-celled. But it's a little bit of a question of definition here. Lots of these shells. They're called tests. Testate amoeba. So I've been reading a little bit in the comments, also with one eye. There's been one question of whether I have ever seen sea monkeys hatch. I've been growing sea monkeys. These are called the Artemia salina. They are called the brine shrimp. But I could not see them yet actually emerge out of an egg. So I did not see that live yet. But I could see, of course, the very young sea larvae. It's not visible on the nose-piece stage camera when you changed the color. That is correct. Okay, good. You know what? Thank you for informing me. So the question is, when I change the color, okay, like this, what am I actually doing? This was the question, right? So you kind of want to know. And so maybe have a look here. I can actually, maybe I'm able to show this to you. I'm able to swing around the, can I do this? Yes, here we go. Do you see this here? There is this knob over here. Do you see this knob? And when I turn this knob, up here, when I turn this knob, then, it changes the color. It shifts something around in the slider, and I can change the color. You know what? Because you were asking, I'm going to take this out, and so that you can have a look of what's in there. Okay. So I'm going to put it back again here. The camera. I used some double-sided tape to stick the camera, to glue the camera directly on the microscope. So I'm going to show you. I'm pulling it out now. Okay. Okay. I'm going to show it. I need now to turn on the desk view again. Just show this to you, how this looks like. Let's put all of the water, everything. It might be dangerous. This is how it looks like. And it's a little bit dusty already, right? And when I basically turn this here, then something in there is kind of moving. Yeah, this is by magnet. It's something in here that's moving slightly. Yeah. Yeah, and this causes the change in color. There's a polarizing filter in here and whatnot. Yeah, a Wollaston prism. Yeah, so this is kind of the thing that I'm turning to change the color. Yeah. So I'm gonna put it back again. Okay, and you know what? So that you can actually see how this looks like. Okay, so I'm putting it back in now. Look at the top again. Can I? The cable is a little bit. Where is this? Here we go. Okay. See? So I can actually slide it in directly like this. And when it's in there, then it locks in place. Yeah, so this is how it goes. Yeah, so let's go a little bit down again with the magnification. Okay, no, well, plenty of amoeba shells, but yeah. So that's a little bit the thing here. Yeah, there's now a little bit. Yeah, the question should I can do the following. I have a whole box of I did not try fairy shrimp yet, but I have those brine shrimp, which are basically salt marine water shrimp, and they come in a pack with a whole bunch of eggs and salt. However, what I found out is you have to be a little bit careful because the salt and the eggs they kind of don't mix very well. So there's a layer of eggs, and then on the bottom or top is a layer of salt. And then when you scoop them out, then you might not get the right amount of salt, right? And that can impact the hatching. How many percent actually hatch? Yeah? This was actually one of the difficulties that I had. Is you have to make sure that the salt concentration is quite right. Okay, so this is kind of the thing. Yeah, I want to move on a little bit because there's still a couple of things I want to show you. I've got a little sheet over here. Yeah, the failed pollen, the exoskeleton of Spiky. Ah, yes, yes. Totally different thing. Change in topic. Yeah, I have to basically forgot to tell you that. Yeah, it goes back to the tutorial about the problem I had with the website. Unfortunately, I had to block, at least for the time being, I had to block some countries because they were basically, I got too many bot attacks. So if you're not able to yeah, basically access the form and the website. Sorry for that. I hope it's gonna be better. But for example, Singapore was one of those countries, unfortunately, that I had to block because I got millions, actually, I think over 2.5 million of attacks in a very short time. Yeah, so this is a little bit a problem. But what I decided to do is the following. I want to show you something. I have not had this in my hands for a very long time. So, and I just would like, and maybe some of you actually know it already. So this is a little bit more for those folks who don't know it yet. I'm going to put all of the water over here on the side, and I don't. Here it is. Okay. I used to back in the day. Look at this here. 2011. I used to I used to feel a little bit frustrated because there were no Microscopy magazines around. So basically, I asked the community at that time to submit articles, and I made magazines. I think I don't know how many I made for several years. I made several editions. You can download them if you want to. And they look. I just today again I had a look at them, and I was quite happy. They looked very nice, actually. I spent quite a bit of time in actually, making the layout of this. Right, this was long before AI. So I had to read every article, and with the best of my knowledge in a spell checker, I tried to edit the whole thing, and then I made a nice layout. Now with AI, it's way easier. And when I looked at this, today, I said, actually, I don't know. It would be kind of nice to have those again. It's a lot of work to do all of the layout, but I can imagine if the community wants, I don't know, maybe we can put out one 4 times a year or something. Yeah. But then again, I know that a lot of things are taking on place now online over the forums and so on and so on. Yeah. So you see that people sent me articles, and I just did the layout and made it available. Yeah. So just I want to float the idea again. I would like to hear your view on this, right? Yeah. And I stopped making it because it was simply a lot of work. And that's the whole thing. Now with AI and having now the possibility to actually do complete AI editing, spell checking, grammar checking, everything, makes it much easier. Okay. So in case you want to have a look at it, you know what? I'm just going to post a link. I'm just going to post a link into the chat. Where is this? I hope that you're able to download it then. Yeah. And in the chat. Yeah, so, yeah. So yeah. So the thing is the following. I'm not. I also don't know. I made the magazine also available over so-called a company called MagCloud. I'm not earning anything here, so the fee that's written on there is basically for 0% profit. So I've not, yeah, because I just wanted to give some. Yeah, does it look familiar? Hey, as we were talking, look, yeah. This is a short article that I made, yeah, and this looks familiar, right? We just looked at this here, yeah. And this is actually a picture that I found on Wikipedia, how the carpet beetle actually looks like, yeah, spiky up close, so to say, yeah. Yeah, so, but that's actually not made by myself, right? Yeah, so just wanted to show this to you, yeah, a little bit. Yeah. So if this is an idea, then yeah. Yeah. Yeah, it's correct. Paper printings have something. Yeah, that's the whole thing. That's what I wanted to have. I wanted to have something. I don't know, solid in my hands a little bit. You already know that a long time ago I did a little bit of astronomy as well. Basically, watching stars and so on. And there were not lots, but several astronomy magazines. But no microscopy magazine. That basically sparked me about 15 years ago. Gave me the idea. Well, actually, we should start one. And that's basically where we are. But I have to also admit there have been some excellent magazines, microscopy magazines, also for amateurs and hobbyists. One of them in German. It was called Mikrokosmos. All of the episodes can be downloaded. They're all in German, hundreds of them, right? But they stopped publishing several years ago. So it's a little bit of a pity. But I don't have the link now, but I know that Mikrokosmos, written with a K, and basically they published hundreds of magazines. It's a traditional microscopy magazine. Goes amateur and also a little bit scientific. Unfortunately, making a magazine is also financially not always sustainable, and for this reason, yeah. Are print copies of the original still available? The thing is the following. Actually, yeah, but I have not really. I don't know if a lot of people actually bought them. It's called MagCloud, and from my homepage, it's like this. From my homepage, there is actually also a link, not only to the magazine, which doesn't work unfortunately, but also to MagCloud. So if you go on the homepage, which is still under construction, into Archive, there's a menu on top called Archive. You can actually go to MagCloud, and you can still buy them. That's the only magazine that I bought. So but it should work. Yeah. Having said that, I still need to fix the homepage, my website. Yeah, so the download link that I gave you should work, but from the main website, unfortunately, I still have to do some tweaking. Yeah. To make it work. Yeah, but this I just wanted to float the idea again. So this was the thing. Then the next thing, yes, also important. Unfortunately, yes, another announcement. Unfortunately, next week I have to skip the live stream again because I'm again occupied. So that you're aware of that. And yes, 4 more things that I would like to show you. Also something that I have not been looking at for a few months now, or almost a year now, and let's see how well this actually works. Be very careful. No, it's not chess this time. Yeah, normally sometimes on Saturday I have to go with my son to a chess competition. Look how dusty it is, but this time it's not. This time it's for private reasons. This here is marine water from northern Germany. I made holidays there, yeah. And this is a little bit I just collected some sand there, and I would like to see if something is still alive, because at least a few months ago, I mean, when was this? 3 years ago or something, I visited northern Germany, and there were lots of diatoms in there, and of course they cannot survive 3 years. But I would like to know if by any chance there is still something visible in there. Okay. The link works good. Okay. At least the link that I gave you. This is you should be able. You have all of the magazines there, and I have to also tell you a little bit now, with a couple of years afterwards, I was not always happy with the quality of all magazines because, as I told you already before, I'm not an editor, right? And for this reason, some of the article qualities might contain typos, spelling, grammar mistakes, and so on. But this is now actually not so much of an issue anymore. So AI occasionally does have its advantage. No, not only occasionally, but in this case, it's quite an advantage. So look what I'm doing. In order to pick up some sand grains, I have to make it a little bit larger here. And let's have a look if this still works. Those sand grains generally are, so not enough sand. You cannot see it because I was too low here. At least a couple of months ago, there were still some. I was still able to find a couple of moving diatoms in here, but I have not very high expectations. But you never know. So in microbiology, you call this a stationary culture. Very stationary. Stationary means when there is no more growth of microorganisms, usually bacteria, then you call it stationary. Normally bacteria, what they will do is they will grow exponentially up to maximum, and when it has reached the maximum, it's referred to as being stationary. And so the difficulty here is we have to really make sure that everything's spread out properly, because those sand grains are fairly large and thick, so high magnification will not be possible here. Oh yeah, so it's still the one from before. I'm just going to add the slide here, and now we're going to look at this marine water. Yeah, and as expected, there's not so much movement going on, but if we're very careful and if we observe carefully, occasionally we are able to see some diatoms. And if I were to now do a time lapse, then sometimes when you speed up the video, then you're able to see actually some of the sand grains moving, because some of the diatoms actually start pulling on it. So still 2.5 years ago, this was a very active culture. Yeah. I'm just reading the comments every time when I'm pausing. Yeah. Have you ever recorded videos while you were away from the live stream? Will you do the same? Are you referring to basically recording videos and then putting them on, what did they call it, Premiere? So it is possible to pre-record videos and then say when they should play, and then the chat is still open. Okay. Yeah, I have done this once. But a little bit of interactivity is missing still. Yeah, I'll think about it. So maybe it depends a little bit on how much time I have. But maybe for next week I could try this again at Premiere, and you'll be able to see it then. Yeah, I'll see how much time I have, and a Premiere basically means that I'm pre-recording a live stream, and I'm just going to play it, and then people can join the live stream, or the Premiere as it's called, and then. You can still use the chat. Yeah. A photomicrography competition for hobbyists. Oh, that's an interesting idea. Like Nikon Small World on your homepage would be cool. Sure. Actually, why not? The thing is the following. If you Google for Nikon Small World photo competition, honestly, the pictures are so good, it's unbelievable. Really. Yeah, I don't know, somebody once also asked me if I want to participate, but I have to tell you, I mean, many of my pictures are kind of nice, right? But they're scientifically not interesting enough sometimes, and those on Nikon Small World, I mean, they're just excellent. Yeah, Premiere is better than nothing. Okay, cool. Yeah. I'll see how much time I have. The reason being that now basically final examinations are starting in my school. I'm a biology teacher, as you know, and I'm also in charge of organizing the final examinations. And for this reason, it's going to be quite busy these days. So this means that sometimes I'll have to stay a little bit longer. I'll see how much time I have. But I mean, just looking at this, there is no movement, right? I mean, the sand grains look nice, sure, all of that, but if there's no movement, it's very difficult to actually see what's there. So that's a little bit the pity. So this kind of looks, the whole thing does look kind of dead. I mean, I don't know if this is one. I'd have to go up. I cannot go up with the magnification very much more because it's so thick. Let's have a look. Because this thing in the center, this long thing in the center might be a diatom. You're going to see in the background occasionally some tiny dots wiggling around. That could be simply Brownian motion. It does not mean that these are cells that are moving. So this is, I don't know if you're able to see this also on YouTube. But this tiny wiggly motion of some of the dots in the background, that's Brownian motion. It's simply a random motion, but there is no active movement. And just to show you somehow the diatoms, yeah. Let me see. Yeah, this one is one. I'm going to turn on the arrow again. Okay, so this one is one, for example. This one over here could be one. Yeah. Interesting idea. Slime mold and phototaxis. Didn't try that yet. I bought myself some slime mold several months ago. I got it to work, but they were growing and moving around happily. But the problem is I could not get them back to life once they were dry. So I'll order some more slime mold. They came in an envelope. Yeah, dried in an envelope. So here we go. Yeah, this here could also be those. You just have to always look at those regular structures. This could be also a diatom. Yeah, but I think it's pretty evident that 3 years is not going to. Yeah, it's not alive anymore. There seems to be a little bit of movement sometimes, but then again, it looks more like Brownian motion. Yeah. Okay. Yes, this is an interesting one. Ever thought about sealing slides and leaving the camera on 24/7 live stream just to let people come and go and watch as things happen? Yes, I actually tried that. Because Ever Smaller was kind of, it's like there are those live streams basically without commentary. You just basically watch what's happening live. And I tried that. And there was, however, a slight problem, a practical problem. I used a sample with nematode worms. The problem is that they were always kind of moving in and out of the field of view. And sometimes for several minutes, you could not see any at all. So this was a little bit the problem. So there was actually, most of the time it looked kind of like this, no movement, and every couple of minutes you could see some movement. So in that sense, it was kind of a little bit boring, but that can be solved actually. The bigger problem was indeed that there was the problem was that the water evaporated, and therefore I had to always add more water every every 30 minutes or so. Yeah. So this is a little bit the problem, yeah. But I tried that. Maybe we can optimize this, should I try that? Maybe. You know what? We tried out. I can show you something. Good. Actually, this might work. Just a second. I found something in here. I'm going to try a different microscope for that. Not with this one over here because of the lack of space on the stage. But look what I have here. Actually, this is a good idea, but I need some interesting microorganisms. Okay, look, the desk view again. This here, yeah, they call it microlife. Okay. I've already used this I think last year or so already. Some of you might have already seen this. It's really delicate. You have to be really careful here. Look at this slide. This is how the box looks like on the inside. There is a pipette with a little bit of some tubing over here to make it airtight, and instruction manual. And what you can do over here is you can actually put a water sample in here, and then the water sample is connected over here, so you can actually observe microorganisms swimming around back and forth here. And there is enough water in here, and it doesn't evaporate, obviously, because you can actually put a cap on here. Yeah. So actually, it would be the ideal solution for such a live stream without a commentary. Okay, so maybe yeah. Some of you already expected it, the old glass enclosure, and you know what? I'm gonna think about this. Maybe it is possible to do some kind of a live stream like this, but in order to make it interesting, there have to be sufficiently many microorganisms in here. So maybe paramecia or something like that. Yeah. So I might have to enrich them, and then I'm gonna put them in here, and then maybe we can observe them. Okay. So I tried to make this also myself, of course, without the glass tubes here. But that was kind of difficult. So you have to cut the glass with a diamond cutter and so on. Yeah. But here it is. Yeah. So I'm gonna move on. Okay. Glycerin from all sides to cover slips, and cyanobacteria inside makes the job. The cyanobacteria do the photosynthesis. Yeah. And the glycerin, or what you can do is. Maybe not just glycerin, but petroleum jelly. This is Vaseline. Vaseline also can be used. Yeah. Okay. Yes, interesting. Trust me, we've been doing it on Twitch for a few weeks. Just a single drop of moss and leave it to see what happens. Sometimes it's exciting. Sometimes it's not. That's interesting. Okay. Yeah. The hope of being able to see something. Okay, so I will do the following. I'll put this back again. Okay, and if I can get it in. Okay, let's do it. Let's put this. That's the instruction manual, by the way, obviously. And so that's number 2. Number 3. I will check now what can the next thing. The next thing that I want to do has been on my agenda now for about half a year. And this is something I have not tried yet, and therefore it's going to be interesting and fun. It's going to be, sometimes I like to put weird things under the microscope. Not sponsored, okay? But in the supermarket I found this here. Okay? This is, I know, a lot of packaging and so on, and plastic, and I get it, right? But I said, well, if there are fruits and banana in here, I wonder what I'm able to find in here. Am I able to find still some cells or not? I really don't know, right? So I decided, well, we're going to find out, okay? So what I'm going to do now is I'm just going to open this thing here. And, yeah. You open it, and then you're supposed to eat it like this, right? I'm not going to eat it like this. I'm just going to, that's of course something. I don't have a, so let's do it like this. And then let's put some of this stuff out here. Does it come out? Whoa. Okay. This is how it looks like. So, and this should be a very easy sample. And I'm going to eat this later. And I just want to know, can I see some of the cells? Right? That's it. You take a small sample. And here, that's already way too much. Way too much. And you take a cover glass and you make a compression sample. Looks like space food, yeah. At least it's packaged like this. Yeah. Will it blend? I remember it. Yeah. Exactly. I remember. Will it blend? They put all sorts of things. I don't know. Even mobile phones under the not under the microscope, but into the blender. That's one of the easiest specimens. Is if you're able to get it really flat and thin. Yeah, on here. And then let's have a look. Okay. So I need to. Yeah. So this is still the sample. Yeah. occasionally also I watch the hydraulic press channel. So in other words, what can you compress? Then there was a will it blend? And now, I don't know. There was also a channel, maybe it still exists, called What's Inside. They used to cut open everything, father and his father-son YouTube channel. And what I'm doing right now is I'm just putting everything what there is under the microscope, and I can already see something here. Yes, it looks like a mess, and occasionally, I don't know, occasionally when these highly processed foods, sometimes I'm really happy if I'm able to find some microplastics, you know. What did I just say? I'm happy if I find some microplastics. Yeah, for example, in artificial meat, I've seen quite a few microplastic fibers. Yeah? So I'm kind of hoping to find again, of course, you know, xylem, because that's very easy to identify. Xylem are the vessels that transport water. Yeah. 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 Yeah, xylem from the banana. Yeah, and I'm kind of worried that this has been maybe mixed too much, but this was a little bit the idea. Is am I able to find? I don't know. The contrast is really. I don't. Let's try the bright field. Bright field is not much better. Yeah. So by the way, what's in here? Apple, strawberry, and banana. So, but for those of you who basically have not seen this yet, of course we can see cells, right? The cells are over here. Let's see the cells here, right? Yeah. So I already know what the next thing is that I would like to do. Of course, we want to find the xylem tissue. I don't know what this is here, but of course, being a natural product, yeah, not surprising. We find all sorts of things here, but it doesn't look particularly nice. It would be kind of cool if there would kind of do some kind of a beautiful thing, you know, or something moving. It's not always like that, but still, if you, I still like to do these things sometimes because simply it's a little bit like chasing and hunting down some structures that look familiar. A little bit disappointing because usually xylem is quite easy to find. I wonder what these long things here are. These are some kind of also plant fibers. Yeah. So what time is it? Just also 52 minutes. So, well, lots of other stuff here. Could be maybe that. Could this be these long things? Not good enough. Yeah. Yeah, this stuff, of course, has been boiled as well. Yeah, so this means that the starch grains of the banana, which is added in here, of course, they are also not quite as easily visible because they absorb the water and they start to become larger, yeah, and swell up. Yeah. Yeah. So, but yeah, good news. I did not see any. Here we go. I did not see any microplastics, at least not to my knowledge. So let's go up here a little bit. Here we go. That's what I wanted to look for, and that's what I found. And now I can basically say we've now also seen this. Let's go up yet more. I have to adjust also the condenser a little bit here. It's too dark. Let's make it brighter here. Yes, this spirally thing here, that is, of course, as you already know, that's xylem. These are basically the cells that transport water, and bananas have large and long. Yeah, here it is. I don't know if you watched my stream last week. If you watched my stream last week, I put some some horse poop under the microscope. One of my students took some horse manure along because she was doing an experiment checking for parasite eggs, and thankfully I could take a small amount home for microscopy for the live stream. And if you put horse manure under the microscope, you're also, of course, going to see quite a few of those. Why specifically those? Because these xylem tissues are quite strong, because they're not only there to transport water, but they also give quite a lot of structural strength to the plant. They're lignified, as we call it. Lignin is wood. wood, right? And so they are indeed physically also stronger. Therefore, they don't break down as quickly, and that's why as easily, and that's why we can see them quite nicely. Yeah. Good point. Good point. Okay. The question is if the job is to transport water, why are they coiled? Wouldn't a shorter path be more effective? The water is not transported in the spiral. So you have to understand it like this: that you have a tube, right? These are the cells, right? Where the water is transported, and the spiral is on the outside, in the cell wall. Okay, basically it's transported in the middle, so to say, right? So the water does not travel in a spiral way, but actually in the middle here. And what we see here is simply the strengthening on the outside. The question is now why is this important? It's actually very important, because if you have a moving liquid or even air or gas inside a tube, the pressure is lower, and then it would collapse. And then it wouldn't be able to move. That's also the reason why we, for example, have here on our neck, we have cartilage rings, right? So also to keep it open, so to say, because as air goes in and out, we have to keep it open, and because flowing air also reduces the pressure, and then it would actually otherwise collapse. Yeah? And we have the same problem with the water. As the water moves up the tube, because of a lower pressure, it would actually collapse together. Yeah, so you can also sometimes see this in garden hoses when you're watering your plants, that if it's not strong enough, then it sometimes collapses on the inside. Yeah. So that's the thing. So it basically is there to keep it open, but at the same time, it's important to make sure that the plant is strong enough, right? Because the xylem gives also physical strength to the stem. Yeah. I've digressed a little bit with my biology here. It's a teacher's disease that I over-explaining everything. Yeah. So, yeah, and sometimes you just, yeah. And those things that you see over here that looks a little bit like folds that you see, yeah, these are actually the cell walls of cells. Yeah. So structural reinforcement, that's correct. Yeah, whole bunch of things here. So, yeah, it's a spreading by capillary action. So, however, what I want to do now is something that just came into my mind. Let me quickly grab my stains. Just a second. It's just over here. Here we go. I've got a whole, and I would like to stain now for iodine, for starch. Yeah. So let me see, where is this here? Look, this is basically. Just want to show this to you here. There's a little bit of my stain collection. What is this here? I already got it here. Those coincidence, iodine. What else? You know the other stains that I have over here. This acetylcarmine didn't work quite well to make chromosomes visible. Not so happy with the results I got with that. Maybe I did not get the concentration right. Methylene blue, according to Löffler. Löffler, 19th century bacteriologist who invented this formulation. Yeah, this is ink, just fountain pen ink. Also works for some things. This one over here is safranin. I used this last week. This is very red color. And this one over here is crystal violet. It's the stain that I'm using right now, also for gram staining and for general purpose staining. Very powerful stain. It stains much more than the Löffler solution. Yeah. But what I would like to do now is the following. I'm going to now put this away again. And we're now going to, I'm just going to use this here. I have no idea. This is actually, it's a good point. I can show you just a second. I hope I have it somewhere. Where is it? Here it is. I bought it over Amazon, and it has the whole label is already turned brown. That's the one that I used. Oh gee, I don't have glasses. 85% water, 10% potassium something, 5% iodine. Okay. It's called Lugol's solution. Lugol's solution is not pure iodine, but also contains some potassium something. And in the meantime, look, it's so aggressive that the rubber bulb over here has turned completely brittle. Yeah, it's completely hard. Yeah. So what I'm doing right now is the following. We're just going to do it the easy way. I'm just going to take a little bit over here, and I'm going to add a bit of iodine, hoping to be able to do a starch test. And then we go again under the microscope, because the starch grains should then be visible very black, and this kind of shows, yeah, basically most like the banana starch. I always like to do it like this because otherwise there's simply too much. Yeah, it does turn pretty black. Okay. It does turn pretty black. So, and one always has to be a little bit careful with iodine because it's quite corrosive. So especially if it touches some parts of the microscope or some metal even, then it likes to corrode. So generally what I sometimes do is the following. I simply try to wash away any excess iodine using simply some water. So here it is. Let's use again the... So I'm going to simply add a little bit of water. Okay. It also makes it a little bit easier to spread, and take a small amount here. Goes on a slide as always. That's more than enough. And cover glass, obviously. Where's the cover glass? Here we go. Yeah. And there's nothing leaking out beneath the cover glass, so that's good. And usually it's also like this, that if there's starch present, then the starch will absorb the iodine anyway. So here we go, and let's have a look. That's a little too. And yeah, and all of those black dots that you see probably contain starch. Yeah. It's a little bit, of course, I added now more water, so for this reason it's a... yeah, here, that's good, that's a cell. Here there were too many of them together. But you will see that there are not many cells that actually contain those starch grains. So I can imagine that maybe due to the boiling process, many of the cells actually broke open. And this starch, of course, started to swell up. Yeah. And yeah, not nice starch, just fragments. This is actually a little bit of an indicator that the whole thing was kind of also maybe, how do you call this, mixed? Yeah, because here, there's at least one intact cell that we have here, or at least looks intact. Yeah. So what I'm doing right now, what time is it? Okay, it's already 1 hour. I'm going to now quickly go through the, how do you say, the chat. It was, I think, 5%. It was basically stock solution of what I bought. Yeah, this was already a question here. Can you add iodine to see the starch in the banana? Yes, that's what I'm doing right now. It's good. But again, this is not quite as nice looking as some fresh banana. Because there you can really see nice starch grains, but it kind of shows that due to the processing, everything was pretty much homogenized. But at least all of those dark patches that you see, that's most likely starch. So let me see. Have you looked at pineapple graphite crystals I found under the cross poles that glow like microlight? Ah, okay. Well, yeah, I'll try this as well. Pineapple. Yeah, they look more like an okay. Phloem isn't that easy to find. Yeah, that's correct. Especially phloem transports are cells that transport substances from the leaves all the way down. Not quite as easy to see because, yeah. And okay. Questions arrive. Thank you. Here, it makes life easier for me. Okay. That's an interesting one. Yeah, I'll read some of the questions here. Does your BX53 beep when it's turned on? Yes, it does, but not very loud. Maybe for a future stream, collect some fresh flowers and take pollen samples to grow and check them at the next week. Maybe, yes, I will try that. I wanted to do this actually already today, but I didn't find not enough pollen. Another off-topic questions I had last week. Did you leave the urine specimen covered or uncovered? I left it covered. However, there was plenty of air inside. I don't have it now, the jar, I have it in the other room. But I basically I left it covered because otherwise, for several reasons, it's going to evaporate, obviously, and it doesn't smell good. There is ammonia. The bacteria in the urine sample, they form ammonia, and this doesn't smell very nice. Yeah? So I mean, that's why I made a few days afterwards. I made a video, and I also did use the pH test strip, and it really turned alkaline due to the ammonia. Have you ever watched sea monkey shrimp hatch under the microscope? I've watched sea monkeys several times already. I think also a few times in this live stream, but I have not actually seen them hatch. So I simply found them after they hatched. Would love to see brine shrimp live stream soon. Yeah, I gotta get some more. I gotta get some more of these brine shrimp because the pack that I have is a little too old. Once they become too old, then the problem is not many of them hatch anymore. Yeah. Put ripe fern. Sorry. Ah, yes. Sporangia drying. I heard that they act like catapults. I think in some fern. I need to explain this a little bit. Could you put some ripe fern sorry under low magnification? A fern on the bottom side of the leaf have so-called sporangia, which basically make spores. And in order to propel the spores, some of them actually they kind of really eject them. Yeah, and this would be nice to see. Yeah. Have you ever recorded videos while you were away from the live stream? Will you do the same this time, or will you take a break? Yeah, I'll try to pre-record something, or I might try the following. I might even maybe just do a non-commented live stream as long as I'm away, and we're gonna just see how this works. Maybe with some music in the background, I don't know. Okay, and then you can join in and you can write comments. So there is still some kind of a community feeling, and maybe even I could write comments, and we can actually observe some of the microbes that are swimming back and forth. However, this only really makes a little bit of sense if the concentration is sufficiently high. If the microbe, if there are not enough, let's say, I don't know, paramecia or so present, then it's kind of not much happens. A competition, that's an idea. I mean, doesn't even have to be a competition. I mean, if people just said, maybe we could do the following. We can, I don't know, set a theme or not, and then people send pictures, and we can. You know what? I'll think about this, and then maybe I can set up also a space on the website where people can upload pictures. Yeah. A premiere is still better than nothing. A premiere is a pre-recorded video which then plays at a certain time. It's still always, be here watching, okay. Just had a thought, could slime mold demonstrate phototaxis, maybe with different wavelengths? I don't know. Never say never, and maybe it also depends maybe on the species of the slime mold. But the question, I don't know. I mean, obviously they do not have any sense organs, but I don't know if they're able to respond to light. Wouldn't surprise me. Biology is so diverse, nothing surprises me anymore. Okay, let me quickly have a look here. A little bit at the bottom. Okay. I'm reading. Okay. How do you recognize microplastics? Okay, that's actually also another live stream that I might want to make. I made one a few years ago. I did collect some... it's called a vegan meat, so which is made of soybeans. So essentially, they used soy beans, and it was processed to make artificial meat. Looks pretty much like meat, but when I put it under the microscope, I found a lot of fibers. And those fibers, in many cases, maybe not in all cases, but in many cases, are actually microplastics. So if you've got synthetic clothing, when you wash it, then in the washing machine water, there are those fibers, and then they don't decompose, and then they kind of go into the drain, and then of course it's going to be it's going to be processed, the whole thing. But ultimately, when it's dumped back into the river after it's been processed, those microplastic fibers are still around because they don't break down. Yeah. So this is essentially a problem, and you can actually see those fibers. Yeah. Is it okay with you? I can throw in the link for the Twitch stream that does. Okay, sure, please go ahead. Yeah, yeah. This is the thing. I didn't even know that there is a 24/7 stream. Yeah, would be interesting. Yeah, I didn't even know that. Okay, yeah. Please, would be. I'd also like to watch it. Yeah, so please feel free to throw in the link into the chat. Yeah. So let's have a look here. What else do we have here? Yeah. Yes, Nile red, that is a hydrophobic stain, and it also starts to fluoresce when it's fluorescing. If you put UV light on it, it starts to glow. I have not experimented yet with this stain. I don't have UV either. Yeah. So yeah. So here we go. Yeah. Okay, that's the same place where we've been already before. Here's another cell. Yeah. So in any case, this seems, yeah. Now we've also put some of this stuff here. Here it is. Yeah. Under the. So I'm going to have to eat this now, because I don't want to no food waste. Yeah. And then as a last thing, let me quickly check terrarium. Yeah, I actually did almost everything. As a last thing, I just want to do the following. In case you were wondering. I go back to the very beginning. I just want to show you how I actually got the pollen, because these things here, where's the desk? And then I'm going to call it a day again. So those are basically the pollen baskets of a bees, right? So this is how they look like. It's almost like, yeah, almost like gravel or so. So the bees basically, when they collect pollen, they will make those pollen baskets, and they're sticking to one of the legs of the bee. And so this is a compressed pollen, and obviously it's not fresh anymore, and maybe that's the reason why it did not germinate. But it's a very nice source of pollen to observe, and what you do is the following. I just grab. I cannot use this anymore, because this one here is contaminated, because I went into the how do you say this thing here? So I'm going to pull it out with a different tweezers, because I have to clean the other one. So let's do it like this. Okay. Then you take a sharp blade. No, this was stupid, of course. Here we go. You just crush it a little bit. Here we go. And you add a little bit of water. Okay, I think I'm gonna think about this. I'm gonna however use a different microscope to do this hour-long live stream, and I have not tried this over a longer time yet. It was just experimental, but I do need a proper source of microorganisms, at least of sufficiently high density, so that people can actually see something. This is way too much water. So what I'm going to do is I'm going to transfer some of this over here. Okay. And maybe I'm going to take a little bit more of this, some more pollen maybe. Yeah, maybe. Yeah. There's going to be plenty. And then I'm going to call it a day afterwards. So, pop, here we go. And let's remove the other slides again. I need to switch over. Yeah. These are basically, so that's basically how it looks like. Yeah. And by adding the sugar solution, I was kind of hoping to actually see them yeah, grow this pollen tube. Yeah, something I have to do a little bit more experimentation. Yeah. Okay, I'm just, yeah. Okay. Yeah, I'm just reading again. Okay. Okay, I'm just reading a little bit here. Yes. It's about the Nile Red. It's about the Twitch stream. Yeah. Okay. Yeah. So that's basically all. What I think what I'm gonna do now is I'm just gonna call it a day. Again, short reminder that next week I won't be doing an interactive live stream, but I'll be thinking about still able to publish something at the same time, so that you have something to watch, and then I'm also maybe able to tune in a little bit on my mobile phone and see how everything is going. Yeah, why not try new things every now and then? And from my side, that's all I want to share with you. Yeah, hope that you liked the live stream, and hope to see you again. Okay, and all the best, and as always, happy microbe hunting. Okay, all the best. Bye bye.